What's up, everybody? So hopefully you did what I said to do in that email on I sent you guys is for you guys to look at these videos, take these notes, work on this stuff. Okay, this right here we're going to work on chapter 7. This is chapter 7, section 4. So chapter 7, section 4 is all on cellular transport. This is incredibly important. This is how our cells they literally get things in and out and transport things through our cells, which is ultimately transporting stuff through the body. So in this, we'll start talking about it, and the next video we will start on chapter 8. So rocking and rolling right through. So diffusion. Particles and solids, liquids, and gases are in constant random motion. This is just an understanding of just life. And all that is just saying that matter is in random motion. It'll move around. And the big thing with this is with it moving around, it does a process called diffusion. And your book's going to define that as the net movement of particles from an area of high concentration to an area of low concentration. Pretty much, it's just a big fancy way of saying what it does is it takes things that is in high concentrations and it spreads it out evenly. Okay, it's kind of like um, if you took your bathtub and you filled it up with cold water halfway, and then you turn the cold water off and you turn the hot water on, and you fill it up the rest of the way with hot. What would happen is at first you could actually feel a difference in the temperatures in the position inside the tub. But if you gave that like 30 minutes and you went back and you put your hand in it, you're no longer feeling a difference. The reason behind that is diffusion. It literally took that energy and just evenly spread it throughout. I know that's more of like a chemistry demonstration, but down here at the bottom, what they have is an example here. Like, let's say I have a beaker and I have some red liquid here and some blue liquid here. And what I do is I have a partition here that's keeping them separate in that beaker. And then there comes a point I lift the partition out and I do nothing to it. What you're going to see is after a certain amount of time, some blue particles are going to move over here and some red particles are going to move over here. And you'll start seeing purple to form where they're mixing. But then there'll come a point to where it's going to completely mix evenly all the way throughout, and it will all be purple. And within this, the reason that this is so special to us is it doesn't take additional energy for this to take place. Diffusion just happens randomly. It already takes place. And this is one big thing that our cells love to use in order to get things in and out of the cell quickly. So this occurs over long enough time, and what it's looking to do is to find equilibrium. In this case, they're talking dynamic equilibrium, and what you'll see with that, what you'll see with this is that it pretty much everything wants to find the state of equilibrium, and that's why diffusion just happens without any energy having to be put in, because everything automatically wants to find itself in this, so it will automatically just do this. Now, in this, the movement of these molecules in these phases does not stop. There's still continual movement. The thing that happens is that the concentration will remain the same. So, like if I took like this blue dot here, if it came over here, there'd be a blue dot from over here, let's say from here, that would go over there. The movement still keeps happening, but it equals out the whole time. Nothing will change that for you. All right, so we use this across our plasma membrane. Remember at plasma membrane what we talked about before here in chapter 7. So this will actually take place across the plasma membrane. A great example of this is water. Water will just freely diffuse straight across. No need. Nothing, no issues whatsoever. It will go completely across the plasma membrane, but most other substances cannot do this. So then they'll use a process called facilitated diffusion. And you remember those transport proteins we talked about when we was talking about the plasma membrane. And I talked about how important those were. This is why it's important. They're used in this facilitated diffusion. And they're used to move these ions or small molecules across the plasma membrane. Now, our cells get very, very creative with this, and we'll talk more about it here in a second, but it's pretty much they get very, very creative with how they can get energy without having to use energy. And you're going to see where some tricky questions can lie here in this chapter, in this section. So diffusion and facilitated diffusions are a type of passive transport. The key to saying something is passive transport instead of active transport, it requires no energy. Okay, it'll just happen. I don't have to do anything. It'll just it'll just take place. I'm not losing energy to it, which is why our cells love it. Not having to use energy is a beautiful thing. So in this, here's how water does this, and this is where typically if we were in class, I actually have this movie, and I pretty much just like to watch it because it's a really good movie. I remember from middle school, and it's one of the main characters in its name is Osmosis, and it's pretty cool. It's the movie's Osmosis Jones. If you guys want to look it up and watch it, go right ahead. Uh, I like it. I think it's a good movie, but some people probably don't. Anyway, so osmosis, all right, this is just how the diffusion of water just takes place over the plasma membrane. So the diffusion of the water across that selective permeable membrane is the process of osmosis. And the important thing to remember about this, excuse me, is that water is the solvent in our cells and the environment. That's why it's so important to stay hydrated. And this is how this thing will work for us. Water will move. It moves across the membrane until the concentration of solutes is the same on the inside and the outside of the cell. Because it's a whole lot harder to move things inside or outside of the cell, inside or outside. Okay, it's hard to move them through because you got a lot more going on. But what we can do easily is just move water and keep the concentration the same. So it's saying like right here before osmosis. So what you'll see, this right side has more ions in it. So what will our cells do to combat that is it will move water from the left side over to the right side, which is why you see more water here. Why? Because it's easier to move that water across the membrane than to move those ions through the cell. Okay? So we're going to talk about three different types of solutions, and these are important. And this is where I really like to talk about and stress the importance of hydration. So first one is an isotonic solution. So this has the same concentration of water and solutes as the cytoplasm of the cell. So this is just saying what's on the inside of the cell and on the outside of the cell is the same. Isotonic is saying it's the same inside and outside. So water still moves through the membrane, but it's entering and leaving at the same rate. Everything's taking place at the same rate. No big changes, no big differences. Now, this is where the differences start coming into play, and this is where it can get bad. The first one is a hypotonic solution. These have lower concentration of solutes than the cytoplasm of the cell. And this is where things can get bad because the net movement of the water is into the cell. So what happens is that lower concentration of solutes that are inside the than that of the cytoplasm of the cell, to even it out, it'll push water into the cell. Now, as water moves into the cell, you can imagine what's happening. I'm pushing more into the cell. So then its greater pressure is building on that plasma membrane. And this is what can cause the problem. Animal cells, they can actually burst when placed in extremely hypertonic solutions, because what happens is so much water is pushed within that cell that it busts. Okay, so that's hypotonic. Hypertonic is the opposite. This is where, excuse me, sorry, I stayed up late watching my football game last night. I was very upset, even though my team was very injured, but it's okay. Anyway, so hypertonic solutions have higher concentration of solutes than the cytoplasm of the cell. So they have higher concentration of solutes than the cytoplasm of the cell. That means there is more outside the cell than there is inside. So the net movement of the water is out of the cell. This is where it can get bad. Excuse me. As water moves out of the cell, pressure starts to decrease, and hypertonic solutions cause wilting within plants. So if you ever watch a plant and you just see it just fall over and wilter and you're like, oh, it's dying, this is what's taking place. The water's having to go somewhere, so to keep it evenly dispersed, what it does is it'll push it out of the cell. Okay. This is, I guess I'll put this out on the internet. It'll probably be funny. This is where I like to talk about anybody that is lactose intolerant. So if you are lactose intolerant, this is kind of like the effect that's happening. If you eat something that has lactose in it, okay, which is a sugar, it comes from dairy products. If you eat that, then what takes place? Your body can't break it down. But the body wants to make sure that it stays in an aqueous or a liquid solution for you guys. Let's say it that way, a liquid solution, that it doesn't become a solid. So in order to do this, it's a very simple process. This right here is what takes place. They take it, and the cells take water and they push it out to make sure that the lactose will stay dissolved and that it can get out of the system, which is why you get the diarrhea when you eat things that are lactose. That's right, ladies and gentlemen. I just said it. So let's move from passive transport and let's start talking about active transport. Now, active transport is opposite of passive. Active transport takes energy. This is how I keep it in my mind. Active activity. If I'm doing an activity, it takes energy. Okay? Excuse me. So sometimes substances, they got to move from lower concentration to an area of higher concentration. In order to do this, to get this movement to take place, we'll do a process of active transportation. Okay, so active transport. And what this does is it takes, the movement of these particles across that cell membrane, okay, against the concentration gradient. What happens is this requires energy, and it is active transport. That's what's taking place. This will occur with the aid of carrier proteins, and they're often called pumps. And we're going to talk about a specific one here in a second, which also is showing a very, very creative way that our cells work. So here's the one that I was talking about, the sodium potassium ATPase pumps. These are found within the plasma membranes in the cells, and it maintains levels of sodium and potassium inside and outside the cell. So an example of this is what happens. So here is this pump that we're talking about. You can see it embedded in this membrane over here. So the first thing that happens is we'll take 3 sodium ions and it'll bond to here. As soon as it bonds, it's then ready for it to do the process it wants to do. So then what happens is ATP, which is our energy source, adenosine triphosphate, that's our main energy source within our cells, comes over here and bonds here to this bonding site. As soon as it does that, it breaks the bond between the third and the second phosphate group and releases energy. As soon as that happens, you can see it's closed like this. So it's closed, but as soon as that happens, then it opens and it closes the bottom where nothing can just freely flow in. Okay, so we're still protecting the cell here. We're not putting the cell at risk, but it opens up. Now, once this happens, the sodium ions, they just flow out. So now I have 3 sodium ions that just float out, and that's outside the cell. Now we'll do a flip process. The second phase to this is getting potassium to come in. So potassium will bond here at these bonding sites. We'll get 2 potassium ions. And then what happens is ADP, adenosine diphosphate, which you see here that came off from the ATP, breaking that bond, adenosine diphosphate comes back. And whenever it comes back, what will happen is it'll release the phosphate from the protein. And then it'll go from this with the potassium in it, and it'll go back to this and let the potassium inside the cell. Once this takes place, then this pump can do it again. And the reason I'm wanting to stress this pump to you so much is what's it doing? It's pushing sodium ions outside the cell. And there's a reason behind that. Whenever you look at it, this is what happens. The pump is not really losing that energy. It's using 1 ATP, but look what takes place with it. What we'll do is we'll build a concentration gradient on the outside of the cell, and we'll do that by pushing these sodium ions out. So the concentration of sodium ions are now higher on the outside of the cell than it is on the inside of the cell. So now what will take place is those sodium ions are going to just diffuse in and just flow straight into our cell membrane, into our cell through the cell membrane. And it does this by using a coupled protein. But here's the really clever thing is in this process of coupled transports, sugar molecules pair up with the sodium ions and enter the cell through the membrane coupled protein channel. This allows sugars to enter through facilitated diffusion, saving energy. In other words, I'm getting an energy source without spending energy. This is why this ATPase pump is so cleverly designed and so important to us. We're getting energy source in, no energy is being used. So the last thing I want to talk about is moving from that sodium potassium ATPase pump is talking about the transport of large particles. These are really big. So what will happen is to transport large particles is a process of endocytosis or exocytosis. Endocytosis is where the cell surrounds an object in the outside environment. You'll see it here. Endocytosis. So what happens, this thing comes in, the cell membrane surrounds it, and then what happens is a portion of that plasma membrane literally surrounds it, engulfs it. And what will happen is kind of like a surgical procedure is there are proteins in our body and our cells that is designed as kind of like they'll make cuts here. When it comes close together, that plasma membrane will seal back together, and then we'll have a vacuole inside the cell that has that large molecule in there, that large particle. But then we also have things that we want to get outside the cell. So let's say I want to get some trash outside the cell. It's a reverse process. So it's like we're adding something to the plasma membrane. So it'll come out like here, you'll see in the vesicle, it'll come up, it'll come right here at that plasma membrane, and whenever it does that, it's like a surgical procedure. What it does is it then takes this membrane and puts it back into the plasma membrane of the cell, and it'll make the cut here, and it'll release this out into the environment. So now it's pushed outside of the cell where we don't want it anymore. So now it's gone. This is exocytosis. The way to keep them in your mind separately, endocytosis, endo. I'm bringing in. Okay, exo, exit. It's going out. Exocytosis. Okay. Hopefully that helps you. And ladies and gentlemen, that is it. We have finished chapter 7. I'm excited to start chapter 8 this week, and we're going to talk about the process of creating energy within cells. If you have any questions, let me know. I will also let you know that we're getting ready to roll out that third packet here. I'm pretty sure we're preparing it, and on Monday we'll be rolling it out. So you'll probably have a video from me yet again on Monday. Hopefully these videos are working better for you guys. I also like the fact that they're here. They're not going to go away from my YouTube channel. So if you need to go back to them to review more, you can. If you have any questions, holler at me on Live Grades. Besides that, peace out, guys.